welcome to the Westin MCAT podcast with your host, Ahmed and
Eric Olson.
Welcome back, everyone, to another episode of the Westin MCAT podcast with Ahmed and Eric. And today we're going to be culminating our three-part series on the Central Dogma of Biology by looking at translation. In previous episodes, we looked at DNA replication and transcription. And today we'll talk about translation, tie all of that together. Before we dive into that, Eric, life as a resident, I feel like I ask you this all the time, but every day it's a new answer. So I'm curious, what do you got for us today?
I don't know if I have much of a new answer for you today.
Okay.
I had a different experience yesterday. Like most of dermatology, at least in an academic center, is like medical dermatology. And so it ends up being like, people with rashes, like, psoriasis and eczema and lichen planus and stuff like that you will learn a lot about in the next couple of months. But then there's like a whole surgical side to dermatology,
right?
That we call surgery, that any surgeon that you ask would say is not surgery, which is fine. It's whatever. And then there's a cosmetic side. And so like, different weeks or different days, we get some exposure to some of those different areas. And, to round it out, there's a pathologic side or histologic side where we look at slides in the microscope, and there's a pediatric side as well. But I had my first like surgical half-day clinic yesterday, which was, I mean, when you're a med student, depending on where you go, but at least my experience was that we got a decent amount of experience like closing surgical wounds. Like, you'll stand in the OR for the whole case and kind of not do much. You might like retract something for an extended period of the case, but otherwise you're kind of just standing there. And then at the end, sometimes they'll say, why don't you put the top stitches in on this wound and kind of close it up. They do lots of stitching in layers, and so the surgeons are doing most of it, or either the attendings or the residents are doing most of it, but then sometimes they'll let you put in a few on top.
Nice.
And so I had a decent amount of experience between medical school and then last year as an intern doing some suturing and things, but it just felt different, like, now that I'm doing it in a dermatology setting. What I had done was like in the OR, and this stitching up the skin was like not a big deal. But in dermatology, like you're maybe taking something out, but a big part of it is kind of the cosmetic of the scar, right? Like, and we concern ourselves with that, where a lot of surgeons are like, I'm fixing something and you're gonna have a scar and it's fine. We're like, we're going to try and take away this like minor defect in your skin, and we're going to try to make it look like nothing ever happened. And so it felt like a little more pressure. It was exciting, like to be a little more hands-on and get my hands in the action sort of. So that was kind of fun.
So would things like lipomas and stuff like that fall under your jurisdiction?
Yeah, absolutely. That's what we did yesterday. We did 2 lipomas and we did 2 like epidermal cysts.
Gotcha.
So that's like general dermatology area, and then different skin cancers, depending on kind of the size and depth and location, can either go to a general dermatologist for a simple excision, or then a lot of times they'll go to the skin cancer surgery specialists, which are the dermatology subspecialists.
I was gonna say, I feel like there's got to be probably a depth past which it moves from derm to like more like niche oncology.
Yeah, exactly. And honestly, I mean, so really the depth at which we would say like, we're not going to do this anymore. And sometimes it'll go to orthopedics and sometimes it will go to general surgery. It's like, once it gets past the subcutaneous fat and into like fascia and muscle, like that's deeper than we go. But even sometimes with like subcutaneous tumors, like there's one tumor that I have done a little bit of research on called the leiomyosarcoma, and like myo meaning muscle and sarcoma meaning tumor, right? So a tumor of the smooth muscle in the skin, and they can arise in the dermis, and they can also arise in the subcutis, in the subcutaneous fat. And it's kind of a gray area. Like sometimes those tumors can be excised by dermatologists, kind of depending on their comfort level, but a lot of times not necessarily because they're so deep, but because they're like more aggressive and they're a sarcoma, like there's potential
Right.
for metastasis there. Then a lot of times they'll go to either orthopedic oncology or surgical oncology. So there is some collaboration. And most academic centers have like what they call a tumor board. And
Yeah,
there's a
yeah, yeah.
tumor board for like every different type of tumor. So there's like a skin cancer tumor board, and then there's like a GI cancer tumor board and a breast oncology tumor board. And you get like a medical oncologist and a radiation oncologist and a surgical oncologist, and a dermatologist maybe in the whatever, depending on the case. And they kind of
Right.
come together in this meeting and like review interesting cases and cases where they need some collaboration and discussion on how to best treat those patients. So, it
Yeah.
is an area of overlap, but we have a lot of support and different people who are helping out with those patients.
It's funny you mentioned the tumor boards. We actually talked about those in a lecture on Tuesday. We had like a medical imaging lecture, and we had this radiologist who is actually a bit of a big deal. Like I want to say did his fellowship at Harvard, did something at Harvard, whether it was fellowship or residency. I think it was a fellowship. And like he's like one of the top radiologists in Canada. Apparently, like you look at his like profile and he's listed at like 17 different websites or 17 different hospitals.
Yeah.
And so he was talking about like the tumor boards and how like bring in like all these different physicians to try to come to a consensus on like the best approach
Yep.
with different we'll say non-routine cases, like things that require a little bit more thought and foresight. And it's cool to see, like, there's a team, obviously there's a team aspect of medicine, but like, I think a lot of times it tends to be very abstract. Like, yeah, physicians work together and, if this one can't solve it, they send it to that one kind of thing. But this was like the first time where I saw like 5, 6 people in a room, like
Yeah.
literally, bouncing ideas off of each other to treat individual cases. You know what I mean? Like that was
And you're right.
opposed.
That's one of the like more concrete examples of like we're gonna get together and have a discussion about
Right.
these patients. And I think there are other instances of that happening. Like we have a clinic that's like co-managing patients with polycystic ovarian syndrome, which is
Okay.
like an endocrine disorder, right? And so you have like the endocrinology team there, and then a lot of times they have dermatologic manifestations, and so the dermatology team is there, and I think there's a pediatrics team in this particular clinic that's there as well. And so we kind of get together and see these patients all at once.
Right.
I think the tumor board is like probably the best example of like we're all going to have a discussion at the same time about this patient.
Yeah. 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 It's
But
cool to see. It's refreshing.
Ahmed,
But
I think that people want to know before we get into our topic. People want I don't know when I was in undergrad, like med school is such a black box to me.
yeah.
To hear what you're up to and kind of what your days are looking like.
Yeah. My days are kind of all over the place.
You're busy, I'm sure.
More my doing than the med school is doing. I'm still like trying to get into a routine. Like, and it's little things like what days am I going grocery shopping? Like, because
Yeah,
like some weeks, like I like buy food that I think will last the week and it lasts the week and then it's perfect. All right, next weekend I'll grab groceries. Some weeks it's like I ran out on a Thursday
and now
and
what? Yeah,
yeah, and like I don't want to eat out just because like I feel bad eating out just because like I try to be like very modest with my money. Like school is expensive, so like, I don't really want to be spending
yeah,
much outside of that. But school's been good. This first course is 8 weeks. It's split up essentially into like 4 2-week blocks, each with a 25% exam. We had our first one on Monday,
how'd it go?
last week.
You
I
feel good
find
about it?
out this Monday.
Oh
Honestly,
man,
man, I'm gonna be completely honest. So for context, like the pass is like a 76. It's pretty high. I'm walking the line. Like I'm close. Like it was like 35 questions, I think, and the pass is 27. You don't have to pass every exam to pass the course, so it's fine.
yeah,
But it was pretty tough. Like of the 35 questions, I'd say like 15 to 20 were just like freebies.
yeah,
But the rest of them, like you had to know your stuff. And I know for a fact I got, I think, 2 wrong at least. So I got 6 to play with. And so
yeah.
we'll see how that goes. I have a feeling a question or two is going to get dropped though, because like talking to like people from previous years, like everyone's like, "Oh yeah, like that first exam we all did so well," and I was like, "There's no shot." Something had to have happened, but a lot of my peers felt the same way. Like it was a pretty tough exam, but fortunately, like this next block of content has been pretty light so far. Um, so yeah, we'll see how it goes, but I'm enjoying it. Like I'm surprised with how much I'm able to do outside of school. Like I've been able to keep up with work for the most, obviously like not working full time, but like I'm working more hours than I thought I would be able to. And then I'm keeping up with, sports and working out and stuff like that. We were talking, earlier off camera how, we've got like a big like first year med student barbecue today that we're really looking forward to. So yeah, I've been enjoying it. There's a lot more self-learning. I don't know if that was something that you felt in med school as well relative to undergrad, but there's a little bit more, hey, like you're required to know this information, like, be ready to be tested on it and stuff provided outside of lecture, which is great because it's less time in class. And it really rewards efficient learning, right? Like you learn things quickly, like you're going to save a lot of time through this sort of modality. But yeah, it's been good. I'm enjoying it. Tomorrow is going to be a very heavy study, catch up, get ahead kind of day, and we're ready for the new week. So, but yeah, so far med school's been good, and yeah, I'm enjoying it.
Sounds awesome. It sounds like you're thriving.
I wouldn't go that far. I'm more towards surviving than thriving currently, but I'm working my way up the spectrum, so we're getting there. Yeah.
Love to
But
hear
in
it.
any case, as for our topic today, we mentioned, wrapping up the central dogma by looking at translation. And I think it'd be helpful for us to just kind of recap what we've already talked about, right? We talked about replication, which, is what allows cells to proliferate and continue passing on genetic information to, future cells. And then we got into transcription, which is how, the information encoded in those genes is essentially exported from the nucleus via mRNA, and then we'll talk about what happens to that mRNA as we go today. But I think even before we dive into translation, it might be helpful just to recap the different types of RNA, because it's not just messenger. Like all of the different types of RNA get transcribed, right? They all are derived from the nucleus, from the DNA. But there's a lot of different types of or a lot of different types of RNA, not just mRNA. I think that's a good place to start, Eric. In your experience with the MCAT, which ones have been, most critical besides the mRNA?
I think you need to know them all in context, and it helps to kind of put them in context of this transcription to translation process. And you make an important point, which is they are all, composed of RNA, and so they're derived from DNA, right? There's a DNA encoding for any of these RNA. The difference to me is that mRNA is kind of in a special category in that mRNA is a means to an end.
Yes. Yeah.
Like you use mRNA to try to generate something else. The mRNA itself is not really doing anything. It's just a code or a recipe for the protein that you're trying to create. And again, M being messenger, right? Messenger RNA. It carries a message to outside of the nucleus. The other two types of RNA that I think are important to know are tRNA and rRNA.
Yeah.
The rRNA, R standing for ribosomal, so ribosomal RNA, again, derived from DNA, and it's then transported out of the cell and is kind of conglomerated with different strands of rRNA and then polypeptides to create a ribosome. And I remember, the first time I learned that, it was like, I think in high school, you learn about ribosomes in biology class. And I think the proposition to me was like, yeah, these are proteins. These are like, this is a protein structure. And then you learn that like, I wouldn't even call it a protein. Like you heard I said polypeptides. Like there are these peptide structures that then interact with RNA strands to create a ribosome. And, I don't think there's too much to know about the rRNA, just to know that it is generated in the nucleolus of the nucleus. And I think, it doesn't play too much into anything else. It's just kind of a factoid to know. And then again, that they kind of come together to make up ribosomes, and then to know a little bit about the function of a ribosome and the structure of a ribosome, which we will dig into as we get into this discussion of translation in a moment.
Right.
The tRNA is maybe a little bit higher yield, or maybe there's a little bit more to know about it, I would say. Again, they're generated in the nucleus from DNA, and they're a little bit different, a little bit more standardized from the rRNA. I think there's some variability to the rRNA. There are a few different strands that act in different ways. The tRNAs, they're always depicted in textbooks as these little like clover leaf shapes. And I think they do that to show that there are areas of base pairing along the tRNA. It's a single strand, right? There's no second strand that's going to become a complement to this sequence. But within a single strand of tRNA, there are different areas that will, bond to itself. And so it ends up having a very characteristic structure because of those segments that are bonded to itself. And I don't know if it really looks like a clover. Again, I think that has more to do with like a 2D representation, but it does end up having kind of like a point at the bottom of it, or on one aspect of it, I guess I should say, because there probably isn't really a top or a bottom. And on the opposite end, and there's a space for binding to an amino acid. And the important points to note about a tRNA, and I think there are like, I'm sure there are specific names for like each arm. I can't remember the names of the different arms of the tRNA, but I don't think that really matters for the MCAT.
Definitely not. No, yeah.
The important points for the MCAT, as far as the tRNA goes, is that there is an anti-codon sequence on the tRNA, and that is kind of this bottom portion, so to speak, of the tRNA. If we'll recall, and we'll get into this in a few moments as well, but the mRNA is composed of 3 base pair sequences that are called codons. An mRNA molecule would be just a structure of, a bunch of amino acids in a row. But the way that it's read inside of a ribosome is in these kind of 3 base pair sequences. And each sequence of 3 base pairs codes for a specific amino acid. And tRNAs are the way that we get our amino acids into the right sequence, based on the recipe that's provided by the mRNA. And so the anticodon sequence will be complementary to the codon. Again, we'll get more into detail of this in a minute. I'm just trying to give an overview of tRNA. But the anticodon will bond to a codon, and so it is each anticodon sequence will carry a specific amino acid that helps to build that polypeptide inside of the ribosome. So I think, again, kind of the important points for tRNA is to know that there's an anticodon sequence, and then there's a space for an amino acid to be bonded to the tRNA. And I think there is a term for the tRNA, correct me if I'm wrong, Ahmed. I think it's called an aminoacyl tRNA.
Yes.
To an amino acid, and there's an aminoacyl tRNA synthetase.
Yes.
Enzyme that bonds those amino acids to the tRNA. I think those are important terms to know as well.
The throwback to molecular genetics in undergrad is you're killing it. I think like I would say that's beyond the scope of the MCAT, but like just think about what those words mean, right? Aminoacyl tRNA synthetase, right? Or synthase. It might be a transferase. In any case, whatever. I mean, like that just that.
So it's an enzyme, right?
Yeah, that there goes to show you like the importance of knowing that detail. But basically, it's transferring aminoacyl transferase, I think. But it's transferring an amino acid to the tRNA. We talked about that clover leaf structure to the top of the clover leaf, right?
The top of the clover leaf, right.
And then the bottom of the clover leaf is, hybridizing with the messenger RNA, right? And so that's our mechanism of delivering an amino acid to this growing polypeptide strand, right? And I think that's the extent to which we want to know this, right? We have a genetic material in tRNA that can bind an amino acid at one end and hybridize with a codon at the other end. Right. And so the 3 nucleotides in sequence from the tRNA that end up hybridizing are what we call the anticodon, which makes sense from our transcription episode. We talked about sense and antisense, right? So you can sort of associate the prefix anti with being complement. Important to note here is just orientation, right? So the mRNA is read 5' to 3'. So your codon on the tRNA needs to be 3' to 5' in order for them to be complementary, right? So if your codon on the mRNA is CAG, right? Fun fact, that's the nucleotide that's repeated too many times in Huntington's. We learned about triplet repeat disorders last week. I thought I would share.
Good stuff.
But in any case, if your codon is CAG, right, 5' to 3', right, your anticodon 3' to 5' would be GUC.
Yeah.
Right? Or 5' to 3' would be CUG. But in any case, the MCAT loves, messing with people 3', 5', messing with orientations and such like that. I think it's really important to keep those ducks in a line. But I think, the other thing to know with the ribosome is just like, where does this tRNA go, right? Where's the protein at any given point? And so you want to think of the ribosome, at least the way that I was taught. And granted, I haven't really seen this come up on the MCAT, but just as an FYI, we've got these 3 sites: the A site, the P site, the E site. E, I remember, was for exit. A was the beginning, the amino acyl site, maybe, and then P, I think, was like peptidyl transferase. Completely irrelevant, but like basically what's happening is this tRNA with the next amino acid is coming into the A site. You have another tRNA in the P site, which is directly adjacent, that has the growing protein. What ends up happening is that entire growing protein gets added to the single amino acid tRNA in the A site, and then that whole thing moves over, and then that's how we kind of elongate this protein.
And it moves over kind of one piece at a time and continues to grow as we add amino
exactly acids. And that actually kind of confused me. I was like, why are we moving the whole protein over just to add one amino acid? It makes more sense for the one amino acid to go that way. But if we think about it from a organic chemistry standpoint, right? We talk about nucleophiles and electrophiles, right? The nucleophile has to attack to attach, right? And so if the amino acid from the one, from the A site moved on to the tRNA and the P site, it would actually release the peptide unless you added it to like the growing end, but that's not accessible because the amino acid is inside. Inside the ribosome. So from a molecular standpoint, it makes sense. From an efficiency standpoint, maybe not as much, but
I mean, from similarly from an organic chemistry perspective, like we talk about proteins being made in an N to C terminus direction. And so if you have, you'd have a carboxylic nucleophile that would have to attack a nucleophile in order to create that bond in the correct fashion, in the correct order.
Yeah. And it's helpful to make that connection as well. It might actually be it might actually be the other way around. It might be the again, this is beyond the scope of the MCAT, but the amino acid in the A site should have a free carboxyl group. And so it would be the amino group of like the growing peptide in the P site that would attack over and elongate that way.
There you go.
Beyond the scope of the MCAT by a wide stretch. But in terms of like the orientation within the ribosome, I think we hit on everything and much more that students will want to know. Just being familiar with the fact that tRNA enters from one site, exits at another, and hybridizes with this mRNA. And I think the last thing I want to mention on this point is sometimes students ask, well, like, how does the ribosome or how does the cell know like which codon is next, right? Like, what if we have to wait a little bit because they don't have the right codon, kind of thing? And it like there's so many of these tRNAs floating around that inevitably the right one sort of inserts. It's not a matter of like, oh, we're waiting for that particular tRNA to get here. It doesn't work that way. There's, at the cellular level, like the number of, particles, tRNAs, mRNAs is enormous. And so that's not really a concern that med students should have.
I think we get tempted to like think that there is some sort of like understanding inside of cells of like what should happen, and like someone has to go and get that tRNA that's going to bond it. But you're right, like it's all kind of up to chance, right? Like there's no thought process on a cell, right? Like things are just happening. And so you'll have like the wrong tRNA with the wrong anticodon coming into that site. It just doesn't have the complementary sequence to the codon, so it will be unable to bind and unable to add its amino acid to that growing polypeptide.
Exactly.
I think it's important to note, we talked a little bit about kind of this aminoacyl tRNA synthetase or whatever we decided it was called. Like that's not happening actively as we are creating a polypeptide structure, right? That has happened in the past. Like we already have all these tRNAs in solution, kind of in the cytoplasm, floating around that are loaded with an amino acid. And so, like you said, there are like, thousands, millions, more than millions maybe of these things that are like floating around in solution and able to come in and bind, but they already all are loaded with an amino acid. And so they're able to come in and just add that amino acid onto the growing sequence. And I agree. I'll just add, the way I remembered those sites, A like is the first letter in the alphabet. So like that's the first site, that's where things start. And then P, I thought of polypeptide. So that's like where the growing polypeptide is going to be sitting, and then use that site, which is pretty straightforward. But I think that can be relevant. And we talk about details on the podcast that sometimes are relevant and sometimes are not. We try to hit the high yield things,
right?
But really, like the more understanding you have of these things, even if it's maybe a little bit beyond what you would need for the MCAT, like these things are going to be helpful for you to understand the processes that are going on. And that will be relevant for you on the test at the end of the day.
Yeah, and I think, we are aware of the fact that our listeners are keen and, are curious. And so we try to cater to that, but really do trust when we say, this is not MCAT relevant. Just so, the last thing you want to do is spend a lot of time learning something that doesn't end up affecting your score even remotely. But in any case, I think the next step, we built this protein now, Eric. We've talked about these tRNAs coming into the ribosome, elongating this growing protein. Eventually, we're going to get a stop codon. A stop codon is basically a tRNA with, depending on your level, you'll see it taught a couple of different ways. Sometimes they'll say, it's just an empty tRNA, and, that breaks out the translational complex and releases the protein. Really, though, there's like a small molecule, I think, that gets introduced, and that disrupts the translational complex. But in any case, the stop codon.
If I may, on the subject of codons, just briefly, sorry to interrupt.
No, of course.
There are, if you think about it, we say that the genetic code is degenerate or redundant. Those mean the same thing. Again, that's probably beyond the scope of the MCAT, but I think the principle is something to know. Like, I don't think you need to know that vocabulary specifically.
Right.
But basically, like, if you think about a codon, right, there are 3 nitrogenous bases in your codon, right? It's a sequence of 3 different bases. And for each space, there are 4 possibilities, A, C, G, and U. And so the number of codon possibilities that there are 64, because we have 4 possibilities times 4 possibilities is 16.
Yeah,
4 possibilities is 64. So there are 64 different like sequences, like AAA, AAC, AAG, AAU, and so on and so forth. Like we obviously won't say them all because there's 64 of them. And how many amino acids are there, Ahmed? I think it's 20, 21, 22, something like that we need to know. Standard.
20 like standard coded for amino acids, and there's 2 other, I just call them weird ones. I never cared to learn what was special about them just because I never needed to, but
I love it. But in any case, the point being, like, there are not 64 amino acids, right?
Correct.
And so when I say that the genetic code is degenerate or redundant, it means that you don't necessarily have one codon for each amino acid, right? You could have
Right.
AAA and AHC and AAG could all code for threonine, and that's not the case. I'm just giving, a possibility of an example.
Yeah.
You could have, for methionine, for example, there's only one codon that codes for methionine. It's AUG, and that's the start codon. That's
Yep.
one that is worth knowing. But for other amino acids, there are multiple different codes or codons that would code for those amino acids. And so it's just a factoid that sometimes will come up, and it's helpful to understand, there are only 20 amino acids that you need to know, but there are all these other possibilities. And so when you're thinking about, so, I've had questions before where they're asking about sequences and they give you a chart. I don't know if they're still doing that, where they would give you a chart with the code of like,
Yep.
here's the codons and here's the amino acids it codes for. And so it's important to recognize, like, there could be different amino acids, or there could be the same amino acid, even if you have different codons. And in the same
Right.
vein, there are then 3 stop codons. So
Yep.
important codons to note are start codon and stop codons. The start codon, again, being AUG. And I don't know if this was just a me thing or if this is an everyone thing, but I would always think of the word augment, which is, not really a word I use in the everyday, but it means
Yeah.
to make bigger or to enlarge. And so that's your start codon. You're augmenting your peptide.
I like that a lot.
And then the stop codons, again, are important to note. Like, sometimes they'll give you a sequence and they'll ask you how many amino acids it will be. And so you might need to know, like, okay, AUG, that's going to be my start. And then you count in 3s until you hit a stop codon. And so what are the stop codons? UGA, UAG, and UAA, I believe. Correct me if I'm wrong.
UGA, UAG, and UAA. Yeah.
Yeah. So, I mean, UGA, like, I grew up in Florida.
I was gonna say, I have a feeling I know where this is
at
going.
University of Georgia. Like, that's what we call it.
Yeah.
I don't know why, like, that means stop codon, but it just stuck in my head. And then
Yeah.
UAG is like you flip the second 2. They all start with U, right? So you have UGA and then UAG, where you flip the second 2 bases.
Yeah.
UAA, I have a hard time remembering, which is why I'm asking you for your help. But
Yeah.
anything to add?
Honestly, not off the top of my head. I think, with these codons, like the yield of them is not crazy high. I'll preface this by saying that. Ultimately, though, I love the augment thing. That's great. And I remember UGA, UAG the exact same way.
Mm-hmm.
And UAA, I guess you just remember. There's actually, you know what though? There's a mnemonic out there that I can't remember off the top of my head, that tells you like all of the stop codons. It was like, trying to remember what it was, but like, you are great. You you are great. You are awesome. And you like something. I can't remember what it was, but there is a mnemonic out there for anyone who's interested. But yeah, really, like, we're getting into the nitty gritty there.
Yeah.
But from a yield standpoint, what's definitely more important, I think, once the translation is done, is knowing what happens next. And that brings us into post-translational modifications, which very different than post-transcriptional modifications. We're not talking about, poly A tails and 5' caps and any of that stuff anymore. We're now talking things like glycosylation and splicing and, what are some of the important post-translational modifications that, you've come across, Eric, with respect to this exam?
I mean, I think, like, I don't think they get into a lot of specifics of like what structures you're adding to proteins.
Yeah,
but I think,
agreed.
glycosylation is certainly one where you're adding like carbohydrate units to different parts of your protein.
Yep.
Lipidation is another that I think is maybe less frequent in reality, but they can kind of anchor a protein to the phospholipid bilayer if you're sending it out to the cell membrane. So that can be important to note.
Right.
I think, I'll give a caveat by saying like the protein structure itself can be hydrophobic. And so you can have like the structure of the protein itself and the amino acids that are making up that protein that can make it hydrophobic and allow it to anchor into phospholipid bilayer. But again, like there are things that the cell will do to kind of ensure or augment different proteins' abilities to do one thing or another. And so lipidation would be one of those. Ubiquitination is one that doesn't necessarily happen like right after a protein is generated. But down the road, you can add these ubiquitin subunits, which kind of marks it for degradation.
Yeah.
And then phosphorylation, I think is one that we should all be familiar with. And that happens serines and threonines. And I think there's one other amino acid where that can occur. Tyrosine. Those are all like available alcohol like OH subunits, right?
Yep.
And I think, we use that as kind of a molecular switch, right? In a lot of cases, I think it can go either way. But in a lot of cases, when you phosphorylate something, you kind of turn it on. And so adding that phosphate group will change the configuration of your protein. And again, all of these modifications, at least in the case of phosphorylation, I guess I should say, will change the configuration of the protein. And that's like really what's allowing it to function in one way or another is the structure determining the function of the protein. And so phosphorylation is one way in which the cell will use frequently to turn on and turn off proteins based on what it needs.
Right. Yeah, and I think the key message there is that these different post-translational modifications serve different functions in cells. Some of them we definitely want to be familiar with, but the big takeaway is just after translation, things happen to give us a wide assortment of proteins, right? I think we may have mentioned this on a previous episode, but the genome, if I remember correctly, is about 19,000 genes, but the proteome is 100,000 proteins, right? So we have 100,000 different types of proteins that we can make from just 19,000 genes. And, this is an important thing to note, major difference between prokaryotes and eukaryotes. We know that prokaryotes can be polycistronic. They can get multiple proteins from a single mRNA. Eukaryotes are not that way, right? And so that begs the question, okay, well then how can we get many proteins from one gene? The answer is these post-translational modifications, which include phosphorylation, lipation, like you mentioned, glycosylation. There's different, there's a word I'm looking for. Obviously, splicing is a big one. There's prosthetic groups that get added. That was the word I was looking for. So lots of things that can happen with a protein after it's translated that gives it a diversity in function. Yeah. But yeah, the specifics of them, the major ones I would know, ubiquitination is a big one. I've seen that a number of times. Glycosylation in the context of, the Golgi is important. Splicing is important. I haven't really seen splice donor and acceptor sites tested too much, so I wouldn't get into that depth, but just being mindful of these different post-translational modifications, where they might occur and what general function they may serve. We talked about phosphorylation, for example, being involved in, activation or inactivation of a protein. Those are the kinds of things that I would know. Make sure you do not confuse them with poly A tail and 5' cap and stuff like that. Those are post-transcriptional modifications.
And if you're like reading the word post-transcriptional versus post-translational, like it looks very similar. So you have to be careful.
Also, I just realized I said splicing. Splicing is post-transcriptional, so completely scrap what I said about splicing. Splicing is not a post-translational modification. Post-translational modifications are, like we talked about, the ubiquitination, the phosphorylation, glycosylation, different additions of prosthetic groups. So lots of different additions that usually, and I think that's fair to say, most post-translational modifications are additions in some form.
Yeah.
At least the ones that you need to know for the MCAT. But
Yeah.
Eric, any cool sort of clinical applications of like post-translational modifications that you've come across?
Post-translational modifications, not really anything that I'm thinking of. No.
Fair
I
enough.
think
I think,
phosphorylation comes up a lot, again, because of kind of molecular switching. And I seem to remember learning a decent amount about ubiquitination as a medical student, although I couldn't tell you why right now. So I haven't seen that yet.
I mean, it's probably I'm speculating here, but it's probably involved in apoptotic signaling, right? Targeting different essential proteins for destruction and degradation. Again, fully speculating, but I would hypothesize it plays a role in like cancer prevention. But again, that's just a hypothesis, no more, no less. But I think, to maybe summarize, right, the entire central dogma that we've run through now over the course of 3 episodes, right? We need to keep replicating DNA so we can create new cells, right? Some cells are mitotically active for life, like our, stem cells of the skin, for example. We're continuously regenerating new skin cells, right? Other cells are not mitotically active at all, like neurons or cardiac muscle, right? Some cells are selectively mitotic, for example, the liver, right? The liver can regenerate, but otherwise it's not mitotically active. But we undergo this process of replication as part of, that S phase of mitosis, right? So that we can make more cells. The transcription translation stuff, that's all in the interphase area of the cell cycle.
Happening all the time,
Exactly.
Yeah.
And so we need that mRNA transcript to export the message from the nucleus to the cytoplasm where the ribosomes can be found, right? Because we don't want to subject that DNA to stress or insult, because that can, alter the genetic code, which alters the function of the cell, etc. And then once we have that transcript, we want to translate it into a functional protein that can then be used to contribute to cellular function.
I mean, can you tell us a little bit? I mean, you mentioned this Huntington thing.
Yeah.
Like, tell us about triplet repeat diseases, because I think that's super relevant in terms of like the molecular biology, central dogma aspect.
Yeah. So Huntington's,
what is Huntington's disease, and what is like triplet repeating, and how does that contribute to the pathway?
Right. So what a triplet repeat disorder is, it's when you have basically a copy paste of a codon many, many, many times. And the reason that's a problem is that our polymerases, our RNA polymerases, and our proofreading mechanisms are not very good at detecting triplet repeats. They're good at detecting frame shifts and premature stop codons and mismatch pairing, but just triplet expansion is not very well recognized. And there's a spectrum, like a certain amount of repeats, not pathogenic. And then there's an intermediate window where it's either mildly pathogenic or potentially pathogenic. And then with Huntington's, I think once you exceed like 55 CAG repeats, CAG being the nucleotide, or sorry, the codon, excuse me, now it's like heavily pathogenic. And so this has clinical applications in the form of like what tests you would use to determine, the genetic cause of somebody's, presentation. There's actually a specific test for suspected repeat conditions, and it's called triplet prime repeat PCR. Basically, it's a test that specifically looks for these triplet repeats. And what's cool, but also kind of scary about these triplet repeat disorders is they have this concept of what's called anticipation, which basically means that the disease gets worse and earlier onset in future generations. Because if you think about the nature of DNA, right, you pass it on to your offspring. If you have 30 repeats, and then as you continue to go through, replication, your polymerases slip and add extra repeats, right? Eventually, you're going to have earlier onset. So when you look at a pedigree and you see earlier onset with successive generations and more severe disease, that's a classic hallmark of these triplet, expansion disorders. Huntington's being one of them. There's another big one that I can't remember off the top of my head, but it's fascinating and it's scary. But I mentioned having a medical genetics unit last week. These are some of the things we talk about, like the different tests you do. What a clinical geneticist does, like how they undergo their detective work to uncover the root cause of somebody's presentation, and sometimes they don't. Sometimes you get inconclusive evidence, and there's for certain conditions like cystic fibrosis, for example, I think there's like close to 4,000 different known mutations, 44 of which are most common. But if you don't have one of those 44, you may have all the phenotypic presentations of cystic fibrosis, but test negative for cystic fibrosis mutant, because these screening tests don't look for every possible mutation. They look for the most ones, right? But yeah, it was really great to get exposed to that field of medicine, because it's not something that I've ever seen. Just the deductive reasoning that takes place to get to a genetic cause if applicable to somebody's condition. But yeah, it's just one of the many tests that clinical geneticists use. They use microarrays and FISH analyses and single gene sequencing, panel gene sequencing. So learning about all those different things and learning what to use and when, and what the limitations are, was actually really interesting. I quite enjoyed it. I didn't like being tested on it, but I enjoyed learning it. And it was cool.
It's familiar though, you already have some familiarity with these sorts of things. And one thing you talked about was like the PCR repeat. What was that called? The testing?
Triplet prime repeat PCR.
Triplet prime repeat PCR, obviously. Yeah, of course. I knew that too. But just like, you know what PCR is. Like you use PCR to amplify a sequence of DNA and test it. So the idea there, is familiar, hopefully. I'm sure it was familiar to you because you know so much about this stuff. But like, you would be looking at a sequence of DNA and your primers are before and after this triplet repeat sequence. And so then you run it on a gel and look at how big that sequence is and look at how many repeats of that sequence you have. One thing that we haven't talked about all too much, but I think is emphasized by this kind of clinical example, is this idea that, the structure of DNA, or excuse me, the structure of a protein will inform its function, right? Like, we talked about this a little bit earlier, but like proteins don't have mentation or cognition. Like they're not deciding that they're going to go and do something in the cell. Like they just do it. And the reason that they do it is because of the way that they're shaped and the other sorts of, molecules or proteins or whatever that can fit into their active site. And so, depending on the way that the protein is structured, which will be informed by its amino acid sequence, that's going to determine how it functions. And so when you start to talk about Huntington's and a triplet repeat disease, like you can imagine, you can kind of envision, if you have your protein that's supposed to work in a specific way and you add all these amino acids in sequence to each other, the same amino acid, 40 or 50 times over, it's like a big loop. Big string somewhere in your protein that's going to change its conformation, change the way that it's able to interact with other proteins or with whatever is supposed to fit into its active site. And so as we have different mutations, and maybe next time we could talk about mutations and different types and how they inform a protein structure, but as we have mutations like this triplet repeat, that's going to change your protein structure. It's going to change your function. And ultimately, in the case of Huntington's, it's going to make it so that your protein doesn't function appropriately. It can't do what it needs to do, and that leads to disease.
Yeah, I think it's important to appreciate, how subtle changes at the level of a protein sequence can lead to tremendous alterations of protein structure, which can have major consequences. And Having that cellular level perspective, even as, a physician who may not work at the cellular level or work at the molecular level, just having a fundamental or foundational understanding of, what transpires at, that, cellular level of organization, I think is important. But it goes to show the importance of some of the things that we learn, right? Clinical genetics is something that interests you. Transcription, translation are not really going anywhere, right? So, widespread applications of the field of medicine, maybe more so than other things on the MCAT, shout out physics. So I think it's a really cool topic. It's a really high yield topic. So hopefully, this series of episodes has been helpful. And students are able to now walk through the central dogma and explain what's happening each step of the way. Eric, do you have anything you want to add?
No, I just agree. This is, if you're going to do clinical genetics, which, is not a huge field, but if you're going to do oncology or radiation oncology in particular, those are areas where this is very relevant. And really, every field has a or some genetic diseases. We have a whole book in dermatology of what we call genodermatoses, which are like genetic diseases that present in the skin. And
Right.
They each have their own intricate and special little mutation that you have to know about and know how it affects cellular function and how that presents in the skin. And I think that's the case for pretty much every field. So
Right.
it is relevant. It will be relevant to you moving forward. It's worth really putting in the time to understand now. And not only will it be relevant for you down the road, but it's, as you said, it's high yield for the MCAT. This whole series in the Central Dogma, I think you'll see it on Chem Phys, you'll see it in biochem as well. And it's something you got to know.
Yeah, I couldn't say it better. I think, yeah, if you guys take anything from today, obviously learn the ins and outs of translation, but really understand it as a cog in this, series of events known as the Central Dogma. Okay? Yep. Happy studying, everyone. Hopefully you guys found that helpful. Thank you for tuning into this episode, and we'll see you guys on the next episode of the Jack Westin MCAT podcast. Take care, everybody.